have our next lecture exam scheduled for Thursday of next week. Okay? And of course, tomorrow, Thursday, another little exam in lab, and it covers through last lab, which is the respiratory volumes and capacities. Yes?
Our next lecture exam is not on respiratory, correct? Just on the cardiac and the blood vessel
It's on
chapter?
chapters 18 and 19, heart and blood vessels.
Okay.
Okay, what we're studying now.
Because our lab has some
I know.
respiratory. Okay, I just wanted to
But
make
the
sure.
third lecture test, which will be the week after that, from now on, if you notice your schedule, you're going to have a test every week.
Yeah.
And toward the end, you're going to have 2. All right? So we're going to have 5 lab tests, 5 lecture exams. 10 tests total, 8 weeks. I think you can figure out that it's going to be tested every week now. Okay? And that next third lab test or third lecture test is going to be the week after the second. So be prepared, which means every day you should be studying. And most of you are. I think you're keeping up. You've got to. Okay? Okay. Today, in fact, starts the 4th week. And the regular semester will be starting the 8th week. So if you wonder why we're going so fast, that tells you right there. Okay? And you're taking this, you can get out of the way quickly. Right? Which means you've got to keep up. Okay. As I said, we talked about the heart. Now you should know the structure of the heart. You're going to trace the blood flow to the heart. You should know the intrinsic conduction system of the heart. You should know the components of the intrinsic conduction system and the pathway of conduction. You should also understand the EKG, the deflection waves of the EKG, what they represent. Remember, it's an electrical representation that you're seeing on the graph. Electrical activity associated with the heart contraction relaxation. That's not what you're seeing. You're seeing depolarization and repolarization, which you should know. You should know that for tomorrow. You may expect to see questions on the EKG. Deflection waves, calculating heart rate, and what those deflection waves mean. Okay? You should also understand cardiac output, which we talked about. Cardiac output is what?
Total volume.
Heart rate and
It's calculated by heart rate,
and
beats per
stroke
contraction per
volume.
minute, multiplied by what?
Stroke volume.
Stroke volume. What is stroke volume?
Amount of blood pumped.
Discharge from one ventricle, either ventricle, so it should be the same, with one contraction.
One
Okay?
contraction.
3 factors affect stroke volume. What are they?
Contractility,
Preload,
contractility,
contractility, and afterload.
Yeah.
Okay? What is preload?
when all the blood stretches the heart, the atrium, get ready to push it through
Not the
the
ventricle, atrium, the
ventricle.
Okay? We're talking about ventricular contraction.
Ventricular.
So preload has to do with the volume of blood that is in the ventricles before they contract. The greater the volume, the more pressure in the walls, the greater degree of stretch, and as you've seen, greater degree of stretch up to a point, the heart contracts more vigorously. That's the way muscle usually responds to stretch, to the point where you overstretch it. Okay? So, with a greater volume of blood, the greater the venous return, greater end-diastolic volume, that's the volume of blood in the ventricle, cardiac contraction, the more force of contraction. You're going to have more stretch. That's the Frank-Starling law of the heart, which you should understand. That's preload. Okay? And again, the difference between what was in the heart chamber, the ventricle before contraction, what's there after contraction, that is your stroke volume. So it's the difference between end diastolic volume, what was there before, and end systolic volume, what was there after. That is stroke volume or ejection fraction. Is all of it ejected? All the blood in the ventricle? No, only about 50 to 60% if the heart is working properly. If it's not working well, it'll be even less. Obviously, you want as much as possible. Okay? What is contractility? Also says deal with force of contraction. What's the difference between contractility and preload? They're both dealing with force of contraction. What's the big difference? Independent of what?
One deals with the
Volume
volume
of blood
of
in the
blood.
ventricle prior to contraction. Has nothing to do with degree of stretch. It has to do with what?
Amount.
Inotropic factors, which you should know.
Positive.
Positive inotropic factors increase contractility. Negative inotropic factors decrease contractility. Wouldn't hurt to know those, okay? So again, it has nothing to do with blood accumulation. It's force of contraction given these factors, okay? What is afterload?
It's left in ventricles.
When they relax.
That's the blood pressure, okay, in the arteries draining the ventricles, when the ventricles are relaxed. That's the pressure that the ventricles have to overcome to force the blood out. Normally, that's not a factor, or it's negligible, in a person with normal blood pressure. When a person who's hypertensive has chronic high blood pressure, it means the heart has to work harder to overcome that pressure, higher blood pressure in those arteries, to force that blood out, okay? So normally when we're talking about cardiac output, we're talking about heart rate and which of these three factors to determine or regulate stroke volume. Preload, afterload, contractility. Which?
Which of those factors determine what?
I said which factors, along with heart rate, basically determine your chronic output in a normal, healthy adult. Preload, contractility, afterload. Contractility. Contractility. Preload is usually fairly stable, okay? Afterload is stable in a person with normal blood pressure. So it's contractility and it is heart rate that really determine your cardiac output. Okay? And again, contractility, force of contraction, it doesn't have to do with what's in the ventricle, the blood volume. It has to do with inotropic factors. So know the inotropic factors, positive and negative. Okay? All right, what affects heart rate? What is the major regulator of heart rate? We talked about that last time. Heart rate, of course, is contractions per minute. What's the major regulator of heart rate? Autonomic nervous system. You got two divisions of autonomic nervous system, sympathetic and parasympathetic. What does the sympathetic do?
Speeds up everything. Speeds it up.
Speeds it up. Because fibers of the sympathetic division stimulate your pacemaker cells in the SA node and your AV node, which of course is what stimulates contraction. Those fibers also stimulate what? What other part of the heart? The myocardium, okay, of the entire heart, including the ventricles. What's the myocardium?
The thick
It's the
muscle.
muscle, it's that middle thick muscular layer. Okay, that's what does the contracting. Right? What's parasympathetic division do?
Slows it down.
It slows the heart rate because again, it's got fibers that stimulate the pacemaker cells of the SA node, pituitary node, the natural pacemaker, and the AV node. How does it affect contractility? More forceful, less forceful?
Less
Why not?
forces.
Because I said so?
Yes.
Your book also says so. Why? I showed you a little diagram up here. It's where the fibers go. Parasympathetic fibers do not go to the ventricular myocardium. They go to the atrial myocardium, but few, if any, to the ventricular myocardium, which means they really have little or no effect on force of contraction. They slow down the heart rate, but they don't affect contractility. Sympathetic does. Sympathetic speeds up the heart rate and gives you more force of contraction. And it's because the fibers of sympathetic go throughout the myocardium, atrial and ventricular, but not the parasympathetic. Parasympathetic has few, if any, fibers to the ventricular myocardium. Okay? Say again?
Through the vagus nerve?
Through the vagus nerve, yes. Vagus nerve 10, not X. All right? And the fibers of the sympathetic, they use spinal nerves. Which ones? Back to ANP1. Remember, it's the thoracolumbar, thoracic, okay?
Which is another name for it,
The what?
for that nerve? Is there thoracolumbar?
That's another name for sympathetic.
Okay.
Another name for parasympathetic as well. I'm sure you've heard of it. Craniosacral. And they use the vagus nerve, which is a cranial nerve. Cranial. And sacral because they use some of the sacrospinal nerves. Okay. But look at that diagram in your book, and it tells you right here. You don't have to memorize it. You look at the pictures, okay? Use your book. You don't read it, at least look at the pictures, all right? Use it more than just a paperweight, all right? You paid good money for that. You might as well use it, yes.
But we also have to know the neurotransmitters.
It wouldn't hurt, you know, the neurotransmitters. That's what accounts for the different reactions, okay? It's neurotransmitters, okay? What's a neurotransmitter of the postganglionic fibers or the sympathetic? Those are the ones that stimulate the heart. Norepinephrine, okay? And norepinephrine is also released by, you're going to see, the adrenal cortex, which we'll talk about later. Okay? And what's the neurotransmitter for the parasympathetic?
Acetylcholine.
Acetylcholine, which also happens to be the same neurotransmitter used by your somatic motor neurons, those that stimulate your skeletal muscle. Okay? So acetylcholine slows down the heart rate. Norepinephrine speeds it up, gives you more force to contract. You can actually drop it on the heart, and you see the heart changing, contracting faster and more forcefully or more slowly. You know what's going on. Okay. Any questions on this? Okay. What else affects heart rate? We said that the sympathetic and parasympathetic autonomic nervous system is a major regulator. What else? Chemicals, yes, various hormones, you should know those. Which ones speed up? Yes?
Did you say something about digestion is flip-flop compared to chemical?
Yeah, and I'll emphasize this when we get to digestive system. I said, overall, the stimulation of the sympathetic of most organs is what? To bring it up to a high state of readiness. You probably heard the fight or flight. That's the overall effect of sympathetic stimulation. Something where the organ is going to do something, the body's going to do something, and use a lot of energy from ATP. Fight, okay? Stand and hold your ground, or flight. You run away. A Monty Python approach, okay? Remember? The Quest for the Holy Grail, did you ever see that? Run away, run away. That's fight or flight, sympathetic. Parasympathetic, what's that? Rest and digest. It brings you down to a low state. Kick back, relax. Except digest. There it's stimulatory. In the digestive system, see, parasympathetic actually stimulates the organs of digestion, and the sympathetic actually inhibits. So it's reversed. That's what I was talking about. Okay? But for now, don't worry about it. Okay?
Yeah, you don't want your digestive system going wild when you're running or fighting.
No. You eat a big heavy meal, do you feel like going out and running a marathon? No. Okay? The exercise usually is push the button of the remote and collapsing on the couch and falling asleep. Okay? What else affects heart rate?
Thyroxine.
That's one of those hormones. Yeah, it's a thyroid hormone, epinephrine, and so on. Okay. What else? Age. Does age have a role? Yes. The older we get, the slower we get, generally speaking. Unless you got a heart problem. Okay. Fastest heart rate is where? In the fetus. Okay. And for the same age, okay, females generally have a little faster heart rate than males. Resting heart rate. Okay. Anything else? Body temperature, higher temperature, faster heart rate, cooler temperature, slower heart. Which is why in some surgeries, they'll cool the body to slow the heart. Sometimes even pack an ice. Okay. All right.
Sounds like fun.
What else? Anything else? How about your physical condition? Does that affect?
Mm-hmm.
The better
Really
shape
great
you're in,
shape,
generally the
slow
lower the heart rate. And trained athletes, especially of Olympic caliber, often they're actually in bradycardia. The resting heart rate is less than 60 beats a minute. Okay?
Why is
That's
that
good.
that?
What?
Why is that that?
Why is that? Because they're basically getting better cardiac output of the shape they're in. They can have a slower heart rate. They got more force of contraction and basically things are healthy all the way around. That's what it's about. Your couch potato's going to be higher. So it wouldn't hurt to know these. All right? Any other questions? Okay, blood vessels. By now you should know the structure of blood vessels. You may just see that tomorrow. Hint, hint. Okay? Find a blood vessel, know the three layers. What's the line of all blood vessels? It's the tunica intima or tunica interna. Internal. What does it consist of? Simple squamous epithelium, single layer flattened cells, liver loose connective tissue. And that's in all normal blood vessels, from the tiny capillaries right up through the major veins and arteries. All right? What's the middle layer? The medium, what's it consist of?
Smooth muscle,
Smooth
thicker.
muscle, elastic fibers. Okay? And that's what's responsible for vasoconstriction when it contracts, it narrows the lumen, and vasodilation when it relaxes. Okay? which adjusts blood flow. Remember we talked about hemostasis, vascular spasm, that's vasoconstriction. Okay, reduces blood flow. Also brings up the blood pressure. Vasodilation increases the blood flow and drops the blood pressure. Okay? And the outer layer,
Endo.
tunica adventitia or tunica externa, and that is what? Connective tissue, collagen fibers, elastic fibers. Their strength and support. Okay? What's generally the thickest layer in a typical artery? Which layer?
Media.
Tunica media, the middle one. And in a vein? Tunica externa. Okay? That's just, think them out. Which you should be able to do. Which is usually bigger, corresponding artery vein?
Artery.
The vein is bigger. Okay? And when you look at it in tissue section, which one holds its shape better? Artery. It's got a thicker wall, it's usually oval or round. The vein would be various shapes and sizes, often larger, but usually partially collapsed, as you'll see. So you should be able to see it. So as I said, from now on, especially when we get to the next unit, digestive system, as I told the morning class, I'll make up for that lack of slides on this one. So those of you who love histology, you're going to like this next unit on digestion, because there are a lot of slides. Okay? And you're going to see a lot of tissue sections, and you're going to see a lot of blood vessels, and also ducts and the salivary glands in the liver and the pancreas and so on. You've got to be able to distinguish the duct from the blood vessel. Blood vessels all are lined with what? The simple squamous, okay? And you can tell because it's a single layer flattened nuclei. But ducts you're going to see are usually a simple cuboidal. How can we tell cuboidal? What's the shape of the cell there? And the nucleus? Usually round. And if it's oval, clumped. Okay? So remember that. It'll help you when you're trying to pick up salivary gland ducts and hepatic ducts and stuff like this, which is coming up. Preview of coming attractions. Okay? Once you get your money's worth. Don't you be bored in here, right? Okay. So you know that, all right? Then we talk about different kinds of arteries. The largest arteries are what? What do they call them? Conducting and elastic. Why? Well, first are conducting the medium-sized arteries. Why are they called elastic arteries? Got a lot of elastic fibers, okay? So they're very resilient. They can expand and recoil, which is good because they act like auxiliary pumps. Ventricles contract, they slam that blood out through the arteries. What happens? They swell temporarily, and then they recoil. So it acts like an auxiliary pump. The arteries are aiding the function of the heart. Okay? That's those large arteries. It's like the aorta, the major branch off the aorta. You should know examples of each of these. Then the medium-sized arteries are the muscular distributing arteries because they have less elastic fibers, but what? More smooth muscle, which means they're capable of what? More constriction and dilation. More vasoconstriction, more vasodilation. And they feed the smallest arteries, which are called arterioles. And it's the arterioles that actually directly feed the capillaries. And it's the capillaries, which are the thin-walled vessels, the smallest, where the exchange between the blood and other tissues occur. Okay? So all these different kinds of arteries, okay, and examples of them, and then the different kinds of capillaries, which we're going to talk about now. Okay? So capillaries, again, are the smallest blood vessels, they have the smallest lumen, and many of them are just barely larger than a red blood cell. You can see. And again, you don't need to memorize these numbers. Okay? You can see 8 to 10 micrometers. A micrometer is 1/1,000,000th of a meter. What's the diameter of a red blood cell?
7.
It's about 7.5 or so. So you can see some of these, the red blood cells can barely get through. As I said, sometimes they'll line up, and then they'll go around a corner, they kind of flex, which is nice because it's a biconcave disc, which means they're more flexible. Okay? They can squeeze through, if you will, these small capillaries. And again, all they're composed of, the capillaries, is that first layer, the tunica intima. Simple squamous, a little bit of loose connective tissue. They don't have the other things associated with them. Now, some capillaries, there's a capillary between an arteriole on one side feeding blood into it, and a venule on the other side. But in most cases, you've got a whole network of capillaries fed blood by the arteriole and drained by the venule. And we call these capillary beds, and that's the city one. So the capillary bed contains what are called the true capillaries. That's a network of capillaries. And then a bypass vessel called a vascular shunt. At any given time, all the capillary beds in the body are not open. They open and close according to the needs of the body. So if you're very active, of course, where's a lot of the blood going to be going? To your skeletal muscles. okay? And you'll be shutting down capillary beds like in the digestive tract and areas like that where you don't really need it. So if you're physically active, you're not gonna be digesting food. It's not a good idea. Can't be having a big heavy meal and then try to go out and run, okay? Do a marathon or whatever, all right? When you're digesting food, obviously you're not gonna be feeding blood to the muscles and vice versa. You're gonna be feeding blood to muscles at the expense of the digestive tract. Okay, the amount of blood to the head is usually uniform, usually constant, although it vary what parts of the brain are being serviced. You're sitting here ozone in class, okay, you may be feeding the brainstem more than the higher thought areas, okay? When you're working out things and stuff like this, and you're getting more of the cerebral cortex, the higher brain activities. Okay, so again, typically we have a capillary bed consisting of a network of capillaries, the so-called true capillaries, and there's a bypass vessel that runs through them, which basically goes directly from arterial on one side to venular on the other. And that's called the vascular shunt. Now the vascular shunt itself has the proximal part which comes off the arterial. This is called a metarterial. Then it's continuous with the distal part, which drains into the venule on the other side. This is called the thoroughfare channel. And then you got this network of capillaries around, which are called the true capillaries. This is your textbook, we just need to look at. All right, this represents the capillary bed. All right, here are the true capillaries. It's a network of capillaries, as you can see. Feeding blood into the capillaries on one side, shown here in red, is an arterial. On the other side, draining those capillaries is the venule, in blue. And of course, as the blood is passing through the capillaries, what's it doing? It's giving off nutrients to the tissues, also oxygen to the tissues, and picking up waste products, including carbon dioxide. So when the blood passes through from arterial to venule, it's usually going to come out on the other side with less oxygen than started with. So the arterioles are going to be carrying blood usually a higher oxygen content than the corresponding venule during the capillary bed. All right, so when the capillaries are open, you can see it's flowing through all the capillaries from the metarterial here through the true capillaries, and then being picked up here by the thoroughfare channel and going right into the venule. However, when you shut that down, you bypass the capillaries, then it goes directly through the vascular shunt from arterial to venule. This is the metarterial. Here's the thoroughfare channel. And like I said, it's just a way to bypass the capillaries. What determines the blood flow? So rings of smooth muscle called these precapillary sphincters. Okay? When the sphincters are open, the blood flows through the true capillaries, you can see here. When the sphincters are closed, like shutting the valve, then the blood flows directly from arterial to venule via the vascular shunt. So it's the smooth muscle here that controls the flow.
So if those tissues don't need any more oxygen, it'll bypass the true capillaries?
Well, if the tissue is not active, okay, these things aren't going to be permanently open or closed, okay? They'll open periodically, then they'll close.
Right.
At any given time, some are going to be shut down and you're just going directly from arterial on one side to venule on the other. Okay? And if it's a very metabolically active tissue, then it's going to be open more. If it's not very metabolically active, okay, like dense connective tissue, like a tendon or a ligament, okay, then it's going to be closed more often. Okay, so it depends on what the tissue is and what's going on, you know, according to the needs of the body. And sometimes following a traumatic accident, what happens? All these things open up, and what happens to blood pressure? It drops precipitously. So, first aid when you treat, okay? You restore breathing, okay? Stop bleeding, and treat for shock. Because what can happen in traumatic injury is these capillary beds open. What this does is cause a precipitous drop in blood pressure. You don't get it up to the head. You can lapse into consciousness and die. And that's why they say, okay, when the victim's there, okay, you're trying to calm them, keep them calm. You don't want them up running around. Make sure you can restore breathing. The breathing is stopped. Stop any bleeding that you can. And have them laying down. Elevate the extremities, particularly the legs. Loosen any clothing around the neck, okay? What you want to do is get more blood to the head to make sure you're getting adequate blood flow. Otherwise, they can go into shock, coma, and death. So you're treating for that. That's because you get a massive opening in these capillary beds, and that causes blood pressure to drop precipitously. Like I said, somebody's going to be closed at any given time for the needs of the body. That's very important. Okay? Again, this meta-arterial peripheral channel, which is the vascular shunt that goes through the capillary bed, is an example of an arterial venous anastomosis. All right, let's look at the different kinds of capillaries. We've already just been talking about capillaries. And you know all capillaries are porous, they're permeable. Because this is where you get exchange between the blood and other tissues. But the degree of which they are permeable or porous varies depending on the type of capillary, as we're going to see. Now, most capillaries are so-called continuous capillaries, which is to say the endothelial cells that line all blood vessels, including capillaries, are pretty much uninterrupted. It's an uninterrupted lining, except for small gaps between adjacent endothelial cells. And these are called intercellular clefts. It's through these intercellular clefts, then, that some things can pass. Small molecules, fluids, and things like this. So small molecules like water can readily pass. Alcohol, which is a small molecule, can readily pass. Okay? But the large molecules don't. And notice that the continuous capillary is the most common type of capillary that we see in the human body. So when we talk about capillaries, most of them are these continuous capillaries. So where do we find them? Very common in the skin. And what part of the skin? The dermis. Because remember the epidermis region? That's epithelium. Typically, epithelium has no blood vessels. Okay, so it's in the dermis of the skin. This is the type of cavity you would see. You'd also see it in most of our connective tissue. Most of our connective tissue well vascularized. A lot of blood vessels. Okay? What's an exception? What connective tissue has no blood vessels in it?
Cartilage.
Cartilage. Yes, cartilage. Okay? Also in our muscles, we see continuous capillaries. We also see these capillaries in the lungs and in the brain. Now the brain, these capillaries are the tightest wall of all. In fact, they don't even have the intercellular clefts. Okay? The cells are tightly adhered to one another with these tight junctions, so they're not very porous at all. Only very small molecules can actually cross. And this is part of the so-called blood-brain barrier. Okay? And what this does is help to keep things from getting into the brain from the blood. So it helps to keep a lot of things out of the brain that you wouldn't want in the brain. But it also makes it difficult to get things into the brain that you might like certain drugs. Okay? Not the happy kind. Okay. Alcohol readily crosses. That's why if you're drinking on an empty stomach, of course, bang, you feel it very quickly. Because first of all, it's readily absorbed through the stomach walls, gets right in the blood, goes right up the brain, and you get the effect very quickly. Which is why, if you want to keep your sense about you, it's a good idea to have something in your stomach beside the alcohol. Eat first, or eat with it. You don't just get blasted right away. But again, very few molecules can actually cross, including a lot of drugs, therapeutic type drugs, because these tight wall capillaries, the so-called blood-brain barrier. So these are these porous capillaries are all in the body, in the brain. But they are a type of continuous capillary. All right, the second type of capillary is more porous than the continuous capillary. These are called fenestrated capillaries. Because in addition to the intercellular clefts of the continuous capillaries, they have what are called fenestrations, which are pores. You can see they vary from about 70 to about 100 nanometers. A nanometer is 1/1 billionth of a meter. So they're small, but what it does basically is give you a greater permeability or porosity to the capillary. So things can cross these capillaries that couldn't cross the continuous capillaries because they're more permeable. So we see these in areas where you need a lot of exchange. So for example, in the kidneys, what the kidneys do, one of the things the kidneys do, and everybody knows that, is it filters waste products from the blood. We're not talking about digestive waste, we're talking about cellular or metabolic waste. So it screens all this stuff out. Okay? Not the particulate matter, but various molecules. So in the kidneys we have fenestrated capillaries. Also in the small intestines, remember the small intestines, you're absorbing nutrients from the breakdown of your food that you digest. You take in these large molecules, proteins, complex carbohydrates, polysaccharides, fats, and so on, and then you break them down into small component molecules, and those are absorbed into the blood through these fenestrated capillaries. So these capillaries are more permeable than the typical capillary. So in the kidneys, in the small intestines, and in many of our endocrine glands. Remember, endocrine glands are the ductless glands. Okay? D-U-C-T-L-E-S, not D-U-C-K. How many times I've seen that? No ducks, quack, quack, quack. No, we're not talking about ducks, all right? All around. Ducts as in tube. Okay? These are the ductless, which means they release secretions into body fluids and usually into the blood. And it's the blood then that carries it to the target areas. And of course the secretions are hormones. So not surprisingly, we have fenestrated capillaries in most of our endocrine organs. Okay, also the ciliary processes of our eyes are part of the ciliary bodies. Those also have fenestrated capillaries. Okay, third type of capillary is the most permeable or porous of all. And these are called sinusoids or sinusoidal capillaries, or if you prefer, discontinuous capillaries. Remember when we talked about the spleen, and you look at the red pulp of the spleen, what are those cavities called? Which the blood percolating. They were called splenic sinuses, but I assume you could also call them what? Splenic sinusoids. Because they are in fact capillaries, very permeable capillaries. So permeable in fact that red blood cells can actually pass across the wall, which they can't in a normal capillary. So these are the most permeable. There are big spaces between the endothelial cells. So big that large molecules, even red blood cells, can cross the walls.
And the lung?
No, not in the lungs. Lungs are the fenestrated, the continuous capillaries. So where do we have these? We have, of course, in the spleen, these fenestrated capillaries. We've also got them,
Liver?
what?
Liver?
Yes. The liver, you're going to see, besides digestive system, the liver has sinusoids, hepatic sinusoids. Other lymphatic tissues, we see them as well. We see them in the bone marrow. Why the bone marrow? Because remember, that's where the blood cells are being formed. So you want to get them into the blood. You have these very porous capillaries here as well in the red bone marrow. Okay? Also in the anterior pituitary, which is one of our hormone-producing glands, our endocrine glands, and in the parathyroid, which is another endocrine-producing gland we'll talk about. So some of our endocrine glands actually have these type of capillaries, like the anterior pituitary and the parathyroid. Others have the fenestrated. But again, these are the most permeable. They tend to be larger than typical capillary, more tortuous because they bend and twist more. What this does is slow down the blood flow. And in many of these organs, like in the spleen and the liver, what are they doing? They're picking out debris from those macrophages. So it gives those macrophages more time to gobble debris up from the blood passing through. And that's helped to filter the blood, as in the spleen and the liver.
It also takes out worn out red blood cells?
That's right. It also takes out worn out red blood cells. You know the spleen did that, so does the liver. And you can live without a spleen, you can't live without a liver. So the liver does also a lot of other important things as well. That's good. Glad you remember that. Okay. So three types of capillaries. Continuous capillary is the most common. They have those intercellular clefts. And of that would be the capillaries of the brain, which are very tight-walled. They're the least permeable. Then we have the fenestrated capillaries, which are more permeable than the continuous in the spleen and the small intestine, many of our endocrine organs, for example. And then the most permeable of all, these capillaries are called discontinuous, sinusoidal, or sinusoids. So permeable, in fact, that red blood cells can cross the wall in large molecules. And again, we see these in the liver, we see them in the spleen, and we see them in some of our endocrine organs, like the anterior pituitary, which is in the brain, or connected to the brain, and then the parathyroid, also endocrine organs as well. Okay. Any questions on this?
So the spleen has sinusoids and fenestrated?
The spleen has these, and so does the liver, and some of our endocrine organs too, like the anterior pituitary and the parathyroid glands, which are not associated with thyroid at all. As I said, in the liver, you have to look at whatever you have, you would assume. These liver sinusoids are the capillaries in the liver, just like you saw in the red pulp of the spleen. And again, they're very permeable, and they're often lined with fixed macrophages called Kupffer cells. But the fixed macrophages are the ones that hang out in a particular organ, usually have a fancy name, also called stellate reticular endothelial cells. And their function, of course, is to pick debris out of the blood passing through. They're filtering the blood. And they will take out microorganisms as well. Okay. So how do materials cross capillary walls? Well, through the intercellular clefts, which they all have, and in the fenestrations, in the fenestrated capillaries, and in the sinusoidal capillaries, also through penocytosis. Remember, penocytosis, the whole endocytosis, where the cell membrane just pinches off little droplets of fluid and takes them into the cell. It's called cell breaking. And then across the endothelial cell membranes. But all capillaries have a degree of porosity. It just varies in porosity or permeability. And the least permeable are those in the brain. The most permeable are those that we see like in the liver and the spleen and some of the endocrine organs, which are the sinusoidal, or discontinuous capillaries. Okay. And also, too, as you'd expect, tissues that are very metabolically active. Okay? Muscles, nervous tissue, kidneys, lungs, liver, and so on, they would have a greater capillary, excessive capillary network within them than those that are less metabolically active, like those of dense connective tissue. So if they're tendons, which basically attach muscle to bone, and the ligaments, which attach bone to bone. And then, of course, some capillaries, some tissues rather, no capillaries. The epidermis of the skin, for example, is avascular, no capillaries, no blood vessels at all. Okay? Cornea of the eye, and the lens of the eye. Why don't you want capillaries there?
Wouldn't be able to see through them.
Yeah, it would impair light passing through. So how do these tissues get fed? Because you've got live cells in the cornea and in the lens. How do you provide these cells with nutrients and take away waste products? If there's no capillaries.
Diffusion?
What?
Diffusion?
Diffusion from what? From the air? No?
From nearby capillary.
Yeah, but what puts it into what? It's not capillary close by, no, otherwise you'd be restricting. There are no capillaries between the cornea, that anterior part of the eye, that's the anterior part, and the lens. There are no capillaries between them. There are capillaries in some of the other tissues. But what fluid bathes the cornea and the lens? Remember the eye when you dissected the eye? In AP1? Yes. Aqueous humor. And that's in the anterior station between the lens and the cornea. Okay? That's what supplies oxygen and carries away carbon dioxide, also supplies nutrients, and
Humor?
takes away the waste. Yeah, the humor.
Okay.
The aqueous, not the vitreous. The vitreous humor is in that posterior segment from lens back to the retina. Okay? That was that jelly-like stuff. When you opened up the eye, that jelly-like stuff that came out, that was the vitreous humor. And the aqueous humor was just the liquid, which you probably didn't notice. Except for the eye. Okay? And two, when they do an eye test, what do? They say, rest your chin on this and look at the little light, and then hit you with this little puff of air. What they're actually doing is seeing the tension on the eyeball, because if that aqueous humor is not drained as it should be, it builds up pressure and results in blindness. And that's called glaucoma. It's a common cause of blindness. If the intraocular pressure, that fluid gets too high, it can cause severe damage that can result in blindness. So when they do an eye test, they test the pressure of that fluid in the eye by actually depressing the cornea a little bit. That little puff of air. Not to be confused with trachoma, which is another disease of the eye, which actually is a bacterial infection. A bacterium called Chlamydia trachomatis, which also is a sexually transmitted disease as well. In fact, it's one of the most common sexually transmitted diseases, called non-gonococcal urethritis. Okay?
You can get it in your eye?
What?
You can get it in your eye?
You can. It's the same bacterium.
Is that why they do the drops in the infants, newborns?
Yes, exactly, because the newborn, especially with a vaginal delivery, may pick up a disease that the mother is carrying, okay? And it could be a sexually transmitted disease like gonorrhea or chlamydia or something like that. That's why they do put the drops. It used to be they put silver nitrate in the eyes. Now I think they just use antibiotics. They put the drops in the eyes to prevent infections of the eye that can be acquired when that newborn is delivered vaginally. Okay, and that's why sometimes, too, they recommend that the mother maybe deliver by cesarean section if she's got an active infection, like herpes or one of these other things, because there's a danger of infecting the newborn. Okay. Okay. And of course, carnage also, as you know, has no blood vessels in it at all. It's avascular. Okay, let's take a look now at the veins. We've looked at arteries, the different kinds of arteries, which you should know, the structure of the arterial wall. We looked at the different kinds of capillaries, which you should learn, examples of each, where they're found, and the different degrees of permeability or porosity. Now let's take a look at the veins. As you're probably aware by now, veins are the blood vessels that return blood to the heart. Remember VA, veins to atria to ventricles to arteries. Veins have thinner and weaker walls for larger lumens than arteries. So overall, corresponding vein and artery, the vein is often a little bit larger than the corresponding artery. Again, they have the same three tunics that we see in the arterial wall: the tunica intima, the lining, the tunica media, and the tunica externa or adventitia. But those layers are thinner. And notice too, overall, veins contain less smooth muscle and fewer elastic fibers. Okay, so not as resilient and not as capable of much vasoconstriction and dilation as the arteries. Okay? And notice too, again, the tunica externa or tunica adventitia tends to be the thickest layer in veins. In arteries, what is it? Tunica media. Okay. Also, our systemic veins and venules are sometimes referred to as blood reservoirs, because when the body's at rest, at least, most of the blood in the body is actually in your veins, these systemic veins, particularly those in the abdominal organs, like the liver and spleen, which are highly vascularized, a lot of blood flow through them, and the dermis, the skin. So when more blood is needed elsewhere, blood can be channeled from these regions and these major veins to other areas of the body. For the rest, about 60% of our blood is in our major veins, particularly in the abdominal pelvic organs and in the dermis, the skin. And again, like I say, when it's needed elsewhere, like say with muscular activity, for example, when you want to feed more blood to the muscles, or with hemorrhaging, major blood loss, okay? And with vasoconstriction in the abdominal organ and veins of the dermis, the skin, then that more blood can be pushed in these other areas as needed. And of course, what causes the vasoconstriction in both veins and arteries is stimulation by the sympathetic division of the autonomic nervous system. It wouldn't hurt to know this, blood volume, okay? Don't need to memorize numbers, but you should know relative volume, okay? Most of it, as I said, at rest would be in our major veins, particularly in the abdominal pelvic organs, the dermis, the skin. As far as the other organs, when the body's at rest, as you can see, systemic arteries and arterioles got about 15% of the blood. Our systemic capillaries throughout the body, about 5% of the blood. The pulmonary blood vessels, which basically service the lungs, about 12%, and the heart, about 8%. By far, though, the majority, some 60% at rest, is in those major veins. Again, particularly the abdominal pelvic organs and in the dermis, the skin. You can see in decreasing amounts. We also have what are called venous sinuses, or sometimes vascular sinuses. And what these are veins that have extremely thin walls. They don't have all the layers or tunics that we see in the typical vein wall. Instead, what they have is the tunica intima or interna, the endothelium, simple squamous epithelium, but they lack the tunica media, and they lack the tunica externa. What they have, though, on the outside is dense connective tissue that supports the wall. And believe it or not, you've already seen examples of this. The dural sinuses, where are they? The cavities between the dura mater, okay, that the blood flows are shown in blue. You should be able to recognize on those head models. And also the coronary sinus. What's a coronary sinus? That's that major vessel, of course, into which the cardiac veins drain, and it in turn drains into what? The right atrium. The right atrium. Okay, so those are specialized veins. The dural sinuses in the dura, around the brain, and also this major collecting vein that we have that basically runs through the coronary sinus or atrioventricular sinus of the heart and picks up or drains blood from the cardiac veins and takes it into the right atrium. Okay. Any questions on this? Okay, just as we saw the lymphatic system, with the major lymphatic vessels, there are valves. And again, they're one-way valves, which helps to basically channel blood toward the heart. Because the blood pressure in these veins, especially the large veins, is very low. So these valves basically, and the fact that these large veins have a very large lumen, the cavity in which the blood flow, this helps basically in the venous return, getting blood back to the heart. The pressure generated by the contraction of ventricles is simply not enough to get that blood back to the heart. So the veins need a little help in returning the blood. By the time you get to the large veins, like the vena cava, the blood pressure is essentially zero. It is extremely low. So it's these other factors that help to push the blood back to the heart. Not the force or not the pressure generated by the contraction of the heart. And there's a picture in your book that shows this. It shows a drop in blood pressure from the arteries. You see systemic and diastolic, and then they kind of come together. And by the time you get to the capillary beds, it's dropped appreciably. By the time you get to the other side of the capillary beds, it's even less. And then, of course, as you get to the veins, you go from smaller veins to larger veins. Because this basically is a divergent system. Okay, unlike the arterial, arteries rather, where it's convergent, where they're splitting into smaller and smaller arteries, here you go from smaller to larger. And by the time you get to these very large veins, the pressure is dropped appreciably. So like I say, the vena cava, it's about zero, or just barely above. The pressure is so low. And that's why venous bleeding is much less serious than arterial bleeding. There is no pulse, and it's just a slow, steady flow from that vessel. But I already know there is a pulse, and that pulse follows ventricular contraction. Okay. Hemorrhoids, you've probably heard of these, sometimes called piles. These basically are swollen, inflamed veins around the rectum. The rectum being part of the large intestine. And what this brings on is prolonged elevated venous pressure. And varicose veins, the same thing. You have these swollen veins. Where you typically see them in the extremities, particularly in the legs, and especially in the lower legs. And this is because the valves can become leaky. What happens then is the blood flows back through the leaky valve, and it tends to pool. And since the wall of the vein is fairly weak to begin with, it causes those walls to expand. And often it's veins that you can see on the surface, and you see this kind of a tortuous network of veins basically, swollen because of the blood, the backflow of blood. This is what we call varicose veins. Okay. And if those veins are around the rectum, those are called hemorrhoids or piles, as they're sometimes known. Now what contributes to varicose veins? The one thing we have no control over. It tends to run in families.
Hereditary.
So you may have a genetic predisposition. So if you got relatives that have varicose veins when they got older, because you see it in older people, okay, then you're more likely to have it as well. Okay, when you're young and born, you usually don't see it. But when you get older, then your body starts to decline a little bit, shall we say. Things don't work as well, including these venous valves. So one thing is heredity. Okay, some people are more prone to develop them later in life than others. Another is, if you're standing a lot, you spend a lot of time on your feet, especially if you're standing in one position, okay? That's why I move around, right? So you're standing at the podium. And if you're on your feet most of the time, at the end of the day, when you're young and immortal, what happens? Your feet start to ache. Why? Because the blood is pooling in the extremities. Because one thing that helps to return blood to the heart is muscle activity. Again, you got the skeletal muscle pump. Same thing, it helps to move the lymphatic fluid through the lymphatic vessels. And it squeezes the vessels. That in combination with those one-way valves help to make sure that blood is going to go back to the heart. Okay, but if you're just standing there, okay, you don't have that muscle activity, the blood tends to pool, particularly in the feet, the ankles, and the lower legs. Okay, so prolonged standing is going to elevate the venous pressure. Obesity, if you're overweight, okay, that also can increase the pressure on those veins, so you're more likely to develop varicose veins. And unfortunately, the latter stage of pregnancy. Regardless of your size, you're going to be bigger, okay? And the body's having to contend with more weight. That puts more pressure on those veins, and you can develop varicose veins. And some women will not have varicose veins until they've gotten pregnant, and then they may have these varicose veins afterwards. So this too can contribute to it as well. And again, anything that we have prolonged venous pressure will contribute to varicose veins. Okay? Pardon me? Yeah, the spider veins, when you have the little superficial veins that kind of round, yeah. Those may not be varicose veins, they're just veins near the surface, superficial veins that you see. They're usually small. Varicose veins usually kind of a tortuous network, and they kind of expand on the surface. I'm sure you see in older people, like myself. But, anyway. Okay, venules. Venules are the smallest of the veins. So they're like the arterioles. Now the arterioles are feeding capillaries, venules are draining capillaries. And the smallest ones consist of the endothelium surrounded by a little bit of connective tissue. The larger venules have the same layers or tunics as the veins. The tunica intima, the tunica media, the tunica externa, or adventitious. Okay? But again, they're thinner walled than the arterioles. Because again, they're handling blood at much lower pressure than the arteriole. As I said before, the venous system is convergent. Small vessels converging, coming together to form larger vessels, which in turn come together with other vessels to form larger and larger vessels. So you can see, as we go from smaller to larger, you have a larger lumen. What does that mean? A lower pressure. And that's why in the larger veins, the pressure is lower than it is in the smaller veins. And by the time, as I said, you get to the vena cava, okay, the superior and inferior vena cava, that pressure is essentially zero, or just barely above. And two, notice that basically the veins anastomose more than do the arteries. So venous anastomoses are more commonly arterial anastomosis. That's where we're having basically these combinations or unions of veins. And because you got a lot more venous anastomoses, a blockage of a vein is usually much less damage and less likely to prevent the flow of blood than an arterial blockage.